Everyone, and welcome to my YouTube channel. So here today we are going to be looking at our A-level biology on the cell membrane. So what are some of the parts and what are the makeup and mechanism of the cell membrane? So if you look at the cell membrane, it is made of a simple structure. So this simple structure has got the head. So this head is called the hydrophilic, meaning to say it's water loving. So our head is hydrophilic, just as we have said. It means to say it is water loving. Hydrophobic, which means to say it fears water, or rather it hates water. So this is how the cell membrane is made. So if we have water this side, the head will make sure that if the head is facing the water, so this is the structure it forms. So let's say this is within the cell, then here it is outside the cell. So within the cell there is also fluid and water will be there, meaning to say the head has to face where water is. So here there will be water. So therefore the cell membrane has got this structure. So now, away from that, we are going to be looking at other parts of the cell membrane. Now if you look at these structures, we are going to be calling them the phospholipids. So these phospholipids are not like just stationary. They move around like this. So we have the structures. They will be moving like that. So they are not fixed. They will move like that. So that is what we mean by the cell membrane when we say that the cell membrane is fluidity. We are just talking about how they are able to move. So now, due to the fact that the cell membrane has got some fluidity, meaning to say when the temperatures are very high, these phospholipids tends to move away from each other. This is where now the cholesterol comes into play. This structure, the cholesterol. So what the cholesterol does is that when it's very hot, it will prevent these structures from more like moving further apart. So that is the main structure of the cholesterol, something like that. So it also maintains the fluidity by overlapping like that to prevent them from moving. If it is too cold, it means to say, just as the principle states, if it is too cold, these would want to move very close to each other. Therefore, what the cholesterol does is that it will be able to stand in between to prevent them to be very, very, very close. So that is the function of the cholesterol. So to maintain the fluidity by maintaining the space between the two or more phospholipids. The cell membrane has got a number of structures. We might also look at these glycoproteins. Glycoprotein is just a protein bonded to some sugar such as glucose. Then we also have the glycolipids. The glucose bonded to lipids is an example. So what these help in its number one, cell signaling and also cell recognition. What we mean by cell recognition, we are simply talking about how do you know that this cell is, let's say, the red blood cell. The red blood cell, you will know it by the presence of the antigen, which is let's say if it's blood group A, you will know it by the A antigen that is found on the surface of its cell. So let's say the antigen will be able to tell you what type of a cell it is. So the glycoproteins and the glycolipids are useful in determining or cell recognition. Cell recognition is what we are talking about, where you can be able to tell what type of a cell it is. So not only that, it also helps in the communication. So cells are identified by the glycoproteins and the glycolipids. The good example I gave were the red blood cells. You can know that the blood group A will have the A antigen. So the A antigen is our glycoprotein, the protein that is found on the surface of the red blood cell, will determine what type of blood group the person has. It also helps communication between two or more cells because they already know what cell this is. We also have this structure which we are going to be calling integral proteins. So integral proteins are just proteins that are embedded within the cell membrane. So now the function of the integral proteins, examples of integral proteins are protein channels. So protein channels allow certain substances to pass with ease. Now remember we said that this is the tail, this is the tail, this is the head. So remember we said that the tail does not like water. Substances can easily pass through, such as small substances such as oxygen are able to pass through, carbon dioxide, these are able to pass through. So therefore, other substances such as water can just pass through here, but due to the fact that the tails of the phospholipids do not like water, remember what we said, they are hydrophobic, meaning to say they don't like water. They do not easily allow water to pass. So therefore, to increase how water passes, water will use what we are going to be calling the channel protein. So the channel protein that water uses, we call them aquaporins. Aquaporins, let me write that. So aquaporins, these aquaporins are just protein channels that effectively allow water to easily pass through in and out of the cell. Now, the only pressure is that there are a lot of substances that dissolve in water. Therefore, they will also need a special way of passing. That is now what we are going to call facilitated diffusion. So facilitated diffusion is simply the type of passive transport in which substances will have to pass through, let's say, channel proteins, so they can pass through here. So now, what is the essence of facilitated diffusion? Not all substances are able to pass through the pores, because many substances, to begin with, they dissolve in water. So since they dissolve in water, already water is not liked by the hydrophobic tail, meaning to say here it would be prevented. So those substances can pass through the cell membrane via facilitated diffusion, where they have to use the channel proteins. So this is basically what our facilitated diffusion is. So remember, facilitated diffusion is passive transport, meaning to say substances will move from a region where there are a lot to a region where there is a lower concentration. So the facilitated diffusion is only there because they are using facilitated diffusion. In the next video, we'll be talking about how ligands operate, how ligand-gated channels operate. See you in the next tutorial. Hope you would like, subscribe, and share for more.